Good afternoon, everyone, and welcome to my presentation. Before I start today's talk, I would like to thank the organizer for this wonderful opportunity for me to be able to present this work at this year Ivan Symposium. Uh, Before I continue, this work will not be possible without Professor Andre Simpson, who allows me to work in his lab and really guide me over the years to become a better scientist and a better person overall. And this work will not be possible without the entire Andre Simpson group as well. So without their dedication, without their hard work, this work would not be possible. And lastly, without our funding partner, particular NSERC and CFI, and also I would like to also thank Broker for their generous contribution over the year to our group as well. Here's an outline of today's talk. First, we want to talk about just a basic about the Benchtop NMR system. And then we're going to talk just briefly about the application of this Benchtop NMR system. And then we're going to really focus on some of the applications, some of the stuff that we can do using Benchtop NMR for environmental sciences, and then final thoughts. Okay, so basic of Benchtop NMR. Here's a picture of how far the Benchtop NMR has come. I mean, way in the beginning, you have very low, ultra low field NMR. What I mean by that is that there is no magnet. You're using the Earth magnetic field, right? The sensitivity is low. There's a lot of work that needed to be done. But with advances in technology, in particular magnetic technology, we were able to push the magnetic field higher to a range where it allows spectroscopy. So what we're focusing on in today's talk will be magnetic field strength that's between 1 Tesla to around 1.5 for a permanent magnet system in this case. And with this magnet configuration, we can now really do some really cool Fourier transform NMR spectroscopy as a result. Of course, over the years, there's a lot of advances in technology, in electronics, right? One of the biggest one of the advances is the pulse sequence in NMR. What I'm saying is that it is now possible to translate some of the pulse sequence at higher field and translate to lower field for better application to improve sensitivity and improve resolutions. And because of the better in terms of magnet technology and the whole higher, you can now have even better sensitivity as well. You have improved sensitivity. It is now possible to acquire, for example, 1D carbon, 1D phosphorus, at a concentration that makes sense. And of course, because of the stability of the magnet, you can now put this magnet at anywhere you want. You can situate it at anywhere you want the analysis to be. And lastly, is accessibility. What I mean by that is that for example, for institutions that want to introduce NMR spectroscopy in their undergraduate lab, but may not have the resources to upkeep a high field NMR magnet. So this is a way to go in terms of bridging this financial gap. I did a brief search on the just keyword in Benchtop NMR in PubMed. As you can see that the number of publications is going up, meaning that there's more and more and more people are using this Benchtop NMR system for a variety of applications. And this leads us to the next topic, which is the application of Benchtop NMR. The Benchtop NMR, have been, there are various applications right now. One of the common application in Benchtop NMR is to look at food adulteration. In this case, we're looking at the adulteration, the authentication of olive oil and honey. And recently, this Benchtop NMR system has been used for reaction monitoring, where it is now possible to essentially sip out samples, aliquot, from your reaction mixture and really monitor the rate of the reaction and the formation of the product. And there's a push of using these Benchtop NMR as a diagnostic tool in hospital setting, where you can look at biofluids like urine and blood to look for biomarkers or start doing disease profiling. And of course, for today's talk, we're going to focus a little bit more on the application of this Benchtop NMR in this domain, that is in environmental science. In order for us to really apply this Benchtop NMR for environmental science, we have to overcome these three limitations. Number one is low signal to noise, just like any NMR spectroscopist, we always face with that. But for Benchtop NMR, it's a particular problem because the magnetic field is really low. The other one is broad line shape. That is due to the fact that the heterogeneity of the sample. And lastly will be water suppression, because for our sample, they're mostly 100% water, and therefore we have to able to suppress the water to really bring out the resonance that is in the baseline. So today's talk, we're going to focus first thing on the how do we improve the resolutions of these benchtop NMR system. Well, one of the way is we look through the high NMR literature, and we found that it is now possible to use these NMR technique where we remove the J coupling. One of the issues with these line shape in one of the instances is the fact that instead of having one peak for one proton, you have multiple peaks for one proton due to the J coupling, right? Higher order coupling and all sorts of these coupling results. So what we want to do is using what we call this pure shift pen to really narrow down these resonances so that we only have one peak for one proton. So therefore, it helps improve the resolution of our NMR spectra. And here is an example spectra here. Here we use the standard JRES and Psyche, and we can see that it is possible, not only does pulse sequence work, we are able to translate some of the pulse sequence that is working on high field for low field application for improved resolution. Another way to improve resolution is spectral editing, right? Spectral editing meaning that we only pick the stuff that we want when we throw away the stuff that we don't want, right? One of the ways we can do this is to look at the different transitional quantum state. As an example, if you have a molecule that have only a singlet, meaning in this case, we call it singlet only, we can have sequences where we select for example, only a singlet. In other instances, for example, where you have a spin network, we can now selecting different quantum transition from double quantum and also triple quantum as well. Of course, that depending as higher quantum state you go, the less and less sensitivity, the less and less sensitive the spectroscopy you're going to get. But having said that, what we can demonstrate here is that it is now possible to really get high resolution NMR spectra using this Benchtop NMR system and applying high field NMR pulse sequence to do what we need to do at a low field NMR, at a Benchtop NMR. So which is great. So we can now really merge two things together and really get to what we want. Now talking about spectral editing, one of the methods that we come up with in our lab is Dreamtime. Dreamtime allows us to select for specific resonances from a mixture, in this case, from a spectral overlapping resonance. Here's an example on a high field NMR spectra where we use Dreamtime as well as Gemstone as a comparison, and we can see that it is possible to select for example, valine and tryptophan, no problem at all at high field, and it has similar sensitivity. But if we were to translate the same sequence at a low field, what we found is that they both technique work, but Dreamtime just a little bit better in terms of selectivity and sensitivity. As you can see here, is that not only are we able to get better selectivity better selectivity where we only select for leucine, we also allow us to select multiple components as well with better sensitivity. So this technique is great in terms of getting the resolution we need for both assignment and also make sure the convolution. And we applied this technique in this case, looking at these C13 labeled diaphnea. What we do in this study is that we have these C13 labeled diaphnea that we culture in our lab. We expose a microplastic, and we look at the changes in the metabolome as a result. They consume this microplastic. As you can read on the news, that this microplastic is a big pollutant in our ocean, in our lakes and river, and we want to see how they impact these diaphnea, which is the aquatic organism, and how they impact the metabolome. And using Dreamtime, we were able to pick out selected resonance that we need to monitor their stress responses. So which is great. Now talking about the advances in technology, in NMR technologies, that now it is now possible to actually do multidimensional NMR on this Benchtop NMR system. Here we're looking at the 2D NMR of pine resin. In this case, we have a standard HSQC. Here we have a different form of HSQC. We call the reverse HSQC, where the CH2 resonances is being flipped onto the axis here, where it is mostly empty. So basically, we use it as much as spectral space as possible to improve the resolution out of 2D. And here is the multiplicity edited HSQC, where you can look at different CH2, CH2, and CH3, depending on their intensity. And here, what we do here also is that we select for CH, CH2, and CH3 system. But having said that, we see that there's some breakthrough in our selection process. So it's not perfect, but having said that, this is a great advancement in terms of really able to get the resolution that we need on a 2D and able to apply a 2D NMR system at a benchtop NMR level with the sensitivity that one's need to actually do some assignments. Now is how do we overcome some of the low sensitivity, right? Low signal to noise. So here's an example. We are currently working on a paper on this. Here is a natural abundance C13 NMR spectrum of 8 white. Here is a surrogate. Here we just want to see, what we can get from it, right? If we only do a single 90-degree pulse, my apology, I forgot to put proton decoupling here. I should have proton decoupling. We do have proton decoupling, okay? We see nothing, right? But if we were to use a new sequence, but apply a sequence called steady state free precession, we actually able to get some signal that coming out. So that allow us to actually think about what kind of pulse sequence we need based on the sample that's given to us, right? So for example, for sample where a single pulse failed to yield the NMR spectra, we can actually use other technique like steady state precession to see can we get can we get signal from that as well? So this is just a preliminary study to demonstrate the feasibility of this pulse sequence on these kind of difficult sample. Now the last thing is water suppression. For environmental science, most of our sample is in 100% water. There's no escaping about it. If we were to freeze dry our sample, dissolve it in D2O, we're actually altering our sample. So now understanding that the bandwidth of these benchtop NMR systems are very low, and therefore our selection need to be really precise and very accurate as well. So therefore we went back to the literature and see, can we find better sequence to do water suppression than what we have currently on the market? Here we did a brief study. Here we look at the different kind of water suppression. We have the aggressive water suppression that based on W5 Watergate. Here we have the standard preset, standard NOC preset as well as purge. And the last graph here is basically looking at the delay in between the binomial pulses and see how they narrow down the suppression bandwidth. What we really want in this case is that we want a very narrow water suppression bandwidth so that it's only suppressing the water and nothing else. But we want to minimize any kind of spillover effect on the side of the water. And what we found is that depending on what you use, you actually can suppress up to 80% of the signal around the water. So therefore, the choice of your water suppression sequence is very important for benchtop NMR system. So here we actually tested out on a urine sample. If we were to just use a standard preset-based NMR sequence, what we can see in this NMR spectra is that it's not as good, meaning that it have a lot of baseline distortion. We're not able to bring out some of the resonances that's in the baseline, right? We still have big residual water that contribute to the artifact. But as we use a more aggressive water suppression, in this case, we use a jump return sequence with a preset here, we can really bring out some of the resonances in the baseline and start to do some assignments. As you can see here, not only JRS is better in every way, it also allow you to get more resonance, get more signal from the metabolite that's reside near the water signal. And for people who are interested in exploring more of this technique, please come to my talk on Wednesday. So final thoughts. With the advancing in pulse sequence and hardware, Benchtop NMR is able to produce NMR spectra suitable for assignment. The portability of Benchtop NMR allows for the spectrometer to be situated at any site of interest. And with the appropriate pulse sequence, one can improve spectral resolution and reduce spectral overlap. And lastly, water suppression can now be implemented with the narrowest bandwidth for Benchtop NMR system. Thank you for your time.
Thank you.
Yes.
And any questions?
Yes. Mm-hmm.
For the water suppression, is it to get the signal through so you don't get a receiver overload, or is it to detect out of little peaks that are next to the water?
The water suppression is twofold. Number one, it allows you to max out the receiver gain so that all the little guys in the baseline it will accentuate. Second thing is that if you don't suppress all the water, what you're going to end up is you're going to have these the residual water will introduce artifact into the baseline, and therefore you have a spectrum that are not faceable. If it's not faceable, therefore there will be introduced issue with quantification at level if you want to continue with that.
Thank you.
Okay, well, thank you.